Hello everyone, my name is Maru, and today I will tell you a little bit about seagrass private life. And by that, I mean microsporogenesis and microgametogenesis. But first, let me introduce the main character of this story. This is the male flower of Syringodium filiforme. This plant belongs to Cymodoceaceae family. It's a dioecious marine species that forms cymose inflorescences. It lacks of perigone, that means no petals, no tepals, so basically the anthers is the flower. And it has 4 pairs of microsporangia. Its distribution is all along the Atlantic Ocean, from Florida to Venezuela, including the Gulf of Mexico. And well, now that you know this character, I will tell you that I decided to divide this talk into 2 questions. So the first one, how are anthers formed? And by this I mean, what happens between having a flower bud and a fully conformed anther? To solve this, I used Scanning Electron Microscopy. And just to give you a really quick background of how these studies are made, I will tell you that there are 4 main steps. The first one, dissection. I remove the bractlets that cover the flower. Dehydration. This was made gradually with different concentrations of ethanol. Critical point drying. What happens here? Well, all the ethanol that is inside of the tissues changes, it goes out and then CO2 goes in. And after this, we can make gold coating. And after that, we're able to see these kind of images. So the first one, this one. It's a non-differentiated flower bud covered with bractlets. It elongates, grows large a little bit. The top of it, we can see it's flat, but suddenly 2 lobes start forming. So this is one and the second one. This flower bud and the 2 lobes then divide again, and so we have 4 lobes. That is 1, 2, 3, 4. And these lobes grow tall. They are prominent now until we have this. 4 thecae. This is clearly 1 thecae, 2 thecae. And the descent line begins to form. After that, it becomes evident. So now we can notice the descent line. The connective tissue is no longer seen, and it grows even longer and takes their normal form, the fusiform, the traditional fusiform form. The filament grows large, and the anther opens. The descent line opens. And we have finally pollen grains, filiform pollen grains, as is shown. And now, the second question. In which moment do pollen grains become filiform? Or are they filiform since the beginning? Well, for this, the 2 main steps are the same, dissection, dehydration, but after that, there's embedding with Eller white. It's a type of resin, and this allows us to create sections. So the tissue looks like this. Remember the first flower bud? The flower bud that was undifferentiated? This is it. So we can see a protodermis that surrounds just an undifferentiated tissue. But afterward, divisions take place. And so these divisions in the primary parietal layer transform into external secondary parietal layer and internal secondary parietal layer. These 2 layers divide in turn, and so we can see here the external one forms what it will become the endothecium and 1 middle layer, and the internal one divides and will turn middle layer, another middle layer, and the tapetum. And this type of development is what is called basic type of development. What happens next? Well, here we have a bigger image of it all. So we can see 4 pairs of microsporangia at a later stage. A little bit of a close-up. And here we see epidermis. So remember that protodermis that was just undifferentiated? It was the same all. Here we can see tannins, these big, massive balls, tannins. And endothecium, which is this one, middle layers, tapetum, and microspore mother cells. What can we see here? So normally, in most of flowering plants, endothecium has thickenings. It plays an important role in dehiscence, but here, no thickenings can be observed. Also notice that the tapetal cells begin to degenerate at an early stage. But what happens next? Those microspore mother cells go into division, meiosis, and so dyads begin to form. These dyads will divide again and will form tetrads, so for a group of 4 cells. And after those 4 cells separate, we get pollen grains. So can you see them? Beautiful, young pollen grains. But what happens with the tapetum? It's gone, or apparently gone, because the protoplast that was inside of the cells has now fused and is surrounding every pollen grain. So if you see here, all this is the tapetum. And what happens with the middle layers? They have flattened. They become flattened. Then, pollen grains begin to grow. They grow large. And as we can see here, the tapetum is still surrounding every cell. And then, every pollen grain is now filiforme, as you can see. And what happens with the tapetum? It's gone. So we can see there's nothing remaining, no remains of the tapetum. And this tells us that the tapetum is plasmodial type of tapetum. Look also that the septum cells are flattened as well, and the middle layers are flattened as well. And a big and elegant way to end the story. This is the anther. We can see that the septum that divided it is gone. The anther wall is broken, and so pollen grains are released, finally released. So well, in conclusion, the main highlights. The anther wall of Syringodium filiforme is comprised by epidermal layer with tanniniferous cells, endothecium with no thickenings, from 2 to 3 middle layers, plasmodial tapetum, a basic anther wall development following Davis classification, and in contrast with Zoster family, pollen grains elongate after meiosis. Thank you for your attention, and I will get questions.
Thanks, very great talk.
Yeah.
Any questions?
No? Well, I hope it's because I'm a great speaker, right?
Why do you call the tapetum plasmodial? Does it move or something, or is it just the shape?
Oh no, it is. So then the protoplasts that are inside the cells of the tapetum. Let me go back. This one here. We can see that the cells are well clearly delimited with a nucleus, and after that, it doesn't exist anymore. So it's called plasmodial because all the protoplasts fuse and it just surrounds the pollen grains. So it's like a plasmodium. There's no clearly delimited, just a lot of material going on there. That's why. what a great speaker I am.
Yeah, no, I can ask you a question.
Yeah.
So in terms of factors that influence anther development, is there much known in that, either environmental or genetic, or could you expand on that?
Yeah, there are a lot of factors that have to do with this.
Yeah.
Genetic, of course. I think there was a talk on the other room about genetic factors, but also environmental factors as well. So, yeah, there's plenty of it.
So if I gave you unlimited supplies of cash, how would you take the research further? What would you like to do in the future?
I think this field of study is, well, non-explored, so there's not much about it. Actually, I think many of the studies have made generalizations. So, for example, this thing that the pollen grain becomes filiform after meiosis was at first generalized for all Cymodoceaceae family, or even for all seagrasses, and that doesn't happen. For example, in Zosteaceae, it happens that the microspore mother cells grow large and then meiosis occurs. So I think that many things, when they are not studied, can lead us to make generalizations that are not correct. So I think what I would do if I had a lot of money is firstly explore the family. I think that that family Cymodoceaceae is not well studied, maybe because it has not many. For example, this genera has only 2 species. Isoetifolium, that is in this region of the Indo-Pacific, and this one, which is just in the Caribbean, in the Atlantic. So I think I will do it that way. First, the family. Yeah.
Any other quick questions?
Thank you.